Hello, my name is Maribel Gendreau, and I teach research methods at Hampton Roads Academy, a K through 12 independent school in Newport News, Virginia. The class is an inquiry-based two-semester science elective offering high school students the opportunity to research a topic of interest in science and technology. Students are encouraged to investigate real-world topic and design, plan, and implement a semester-long investigation to address a research question. During the first semester, the course explored various topics such as research methodology, ethical research practices, biotechnology, and data analysis tools. The second semester, students work towards the completion of a research project culminating in a final research paper and oral poster presentation. After the course, students will have the option of taking the biotechnology assistant certification exam. In this course, students also learn basic skills essential to conducting biotechnology research. They're introduced to microbiological and cell culturing techniques, as well as DNA and basic protein analysis techniques. They also learn to keep an accurate account of their experiments in their lab notebook. By the end of the course, students understand different scientific research designs and methods. They learn how to set up a research studies, perform literature reviews, and refer to and cite from scientific literature. Each quarter, students select a topic in affecting the US or world population, review the literature, and write a persuasive paper expressing their opinion, which they will then defend in a Socratic seminar-style presentation. In addition, they read A Cracking Creation, Gene Editing and the Unthinkable Power to Control Evolution, and reflect on the topics learned through discussion boards. Isabelle is an 11th grader interested in pursuing science in college. Her experiment stems from a study she read on how prescribed antidepressants can increase antibiotic resistance in bacteria.
Hello, my name is Isabelle and I study the difference in the effects of prescribed and natural antidepressants on the resistance of E. coli to antibiotics. The prescribed antidepressant tested was sertraline, and the natural antidepressants used were lavender, omega-3, and 5-hydroxytryptophan, abbreviated as 5-HTP. The effects of these antidepressants were tested using amoxicillin, ciprofloxacin, and erythromycin. This experiment had two negative controls: Luria broth with no bacteria and E. coli cultured in Luria broth. The positive control used was sertraline-treated bacteria, a commonly prescribed antidepressant. It was hypothesized that natural antidepressants would not increase bacterial resistance to antibiotics as much as prescribed antidepressants, and that bacterial colonies would not undergo any significant morphological changes. This experiment proved that natural antidepressants increase the resistance of E. coli to antibiotics as much as prescribed antidepressants do. However, its effects depend on the type and concentration used. In addition, bacterial colonies from treated cultures did not undergo any morphological changes besides a shift in form. Many people are taking prescribed antidepressants now that there is more awareness on the importance of mental health. However, studies have shown that antidepressants increase bacterial resistance to antibiotics, making it harder for patients to recover since the antibiotics are not as effective. Therefore, I conducted this experiment to find out whether natural antidepressants increase bacterial antibiotic resistance as much as prescribed antidepressants do. Three natural antidepressants, lavender, omega-3, and 5-HTP, along with one prescribed antidepressant, sertraline, were prepared at different concentrations according to Table 1. Non-pathogenic E. coli was cultured, and 100 microliters was added to each of the LB broth tubes containing different concentrations of antidepressants. All tubes were incubated overnight at 37 degrees Celsius. LB agar plates were streaked for single bacterial colonies the following day using a sterile inoculation loop. A negative control plate with no bacteria was included in this investigation to ensure no contamination of the plates. In addition, a second negative control plate with bacteria not grown in antidepressants was used as a comparison point. The morphology of the colonies were classified based on form, elevation, and color, according to Figure 5. Three antibiotics were used for the Kirby-Bauer test: amoxicillin, ciprofloxacin, and erythromycin. Antibiotic discs were placed in quadrants 2, 3, and 4, ensuring each disc made contact with the agar. The first quadrant was used as a control with a sterile paper disc containing no antibiotic. Plates were then incubated for 24 hours at 37 degrees Celsius. After 24 hours, the zone of inhibition was measured and compared with the controls. The results of this experiment did not support the hypothesis that natural antidepressants will not increase bacterial resistance to antibiotics as much as prescribed antidepressants do. The results suggest that sensitivity to antibiotics depends on the type of natural antidepressant used and its concentration. In Figure 1, the sertraline-treated E. coli shows increased resistance to amoxicillin and decreased resistance to ciprofloxacin compared to the negative control. In contrast, Figure 1 shows lavender-treated bacteria showing the reverse, with the least resistance to amoxicillin and an increased resistance to ciprofloxacin. In Figure 2, the omega-3 treated E. coli results were similar to those of the untreated E. coli, with only a slight decrease in resistance to ciprofloxacin and a slight increase in resistance to erythromycin. Similar results were also found in 5-HTP compared to the untreated E. coli, as seen in Figure 3, with only a slight decrease in resistance to ciprofloxacin and erythromycin, and a slight increase in resistance to amoxicillin. Overall, the E. coli was generally most resistant to erythromycin, which is a widely used antibiotic to treat skin conditions and chest infections. The bacterial colonies did not undergo any significant morphological changes. As seen in Figure 6, sertraline-treated E. coli were the only circular colonies. The remaining colonies were punctiform. The lavender-treated E. coli produced the smallest punctiform colonies in medium and low concentrations. In contrast, omega-3 treated E. coli and 5-HTP treated E. coli produced similar-sized colonies to the untreated E. coli. Further studies to understand the effect of natural antidepressants include testing other widely used antibiotics such as tetracycline to produce a broader range of results, testing a wider range of concentrations, and including more samples for accuracy. Additionally, the morphology of bacterial colonies could be studied in more detail to see if there is an evolutionary component in bacterial characteristics beyond their size, color, and texture. By contributing to the findings on the effects of natural antidepressants on bacterial resistance to antibiotics, I hope to bring forth new data that could potentially contribute to developing an alternative treatment for patients suffering from depression without risking the treatment of other common bacterial diseases. I would like to thank Dr. Gendreau for giving me the opportunity to learn these new skills and for guiding me through my research process. Thank you.